This is a production of Cornell University.
So this is a seminar for me to re-up my adjunct status. I am a research geneticist plants with USDA-ARS. And I think everybody knows me, so I'm not going to give any details from my CV or whatever. You know, the faculty members will receive my CV as well as a statement of interest, and you'll get to vote on me. Since we're here in person, I failed to do this earlier last year and this year, which is to present the Murphy-Munger Awards. And so there are actually 3 people up. I'm not sure if they're all on Zoom with us now or here in the audience, but let's give them a round of applause. Congratulations. And yeah, it turns out I have too much information to give you, and so I'm just going to be barreling through. You know, there'll be little interludes. I have 4 little vignettes, so maybe 1 question or 2 at the end of each vignette. But so yeah, these are some of my quantitative things. They make me happy. So prediction of metabolites, additive relationship matrices for sugar kelp that has a biphasic life cycle, not just genomic selection, but genomic mate selection. And then the last thing that I'll spend a little more time on is this Bayesian optimization of breeding scheme budgets. Okay, so the first one, they're not really my quantitative things. They're definitely, it's work that's being pushed forward by various postdocs and research associates. Oats are a known healthy crop, and so this is in part due to secondary metabolites, and so breeders are interested in being able to manipulate these and the levels of these, and so we've been looking into that with Mike Gore, and the main person responsible for what I'm going to show you here is Lauren. So here's the premise of what we're doing. The idea is, okay, so you have some metabolite, there are variants that cause it to have different levels. If you have a handle on the causal loci, when you're estimating their effects, you want to estimate those effects from a different distribution than loci for which you have no evidence of causality. I thought Malachi Campbell, stated that really nice in his paper last year. And so the hook that we're using here is when you do untargeted metabolomics, you get a lot of data, and we want to try to use that data more completely to sort of prioritize loci according to their probability of being causal. There are different possible approaches. We have a discovery panel. We have metabolomics there. We could match 1-to-1 between metabolites in the discovery panel and metabolites in the prediction set. The thing that we're actually going to try to do, though, is combine a series of metabolites in the discovery panel on the assumption that by joining information from them, we'll get a higher ratio of signal to noise, I guess, for this prioritization. Then the other part is actually, once you've decided which loci you're interested in, is estimating their effects. We could estimate them directly in the discovery panel. We're not doing that. We're using just the discovery panel for this prioritization, and then we're estimating effects in a downstream prediction panel. So this is what this looks like, is sort of these different possibilities, and we're in that lower right-hand corner. Okay, so how do we actually draw effects from different distributions? If you do, let's see, can people see this? If you do standard RR-BLUP, you're drawing all of the effects from the same distribution. It's a normal distribution, so alpha are the effects of the loci. It has 1 variance for all effects. And we're using a fairly simple approach. We have loci that we have some evidence that they're causal. I'm putting a subscript C for those, and we're pulling them from 1 normal distribution. It has a different variance than the distribution of the loci that are the rest of genome loci, so for which we don't have evidence of causality. And in principle, this variance component, sigma squared C, should be bigger than the variance component, sigma squared R, right? So what Lauren has done is, in the discovery panel, to do GWAS on all of the metabolites, we have some 1,600 metabolites. And then she brings together SNPs that are hit in different ways. Some sets of SNPs are sort of general, you know, across all of the metabolites, a SNP is hit, and we choose SNPs that look important because they're hit more than once. In other models, we're bringing together SNPs that hit metabolites of particular structures, so phenylpropanoids or lipids. And then in a third set of models, she brings together SNPs that participate in metabolites that are part of the same pathway. So terpenoid pathway, mevalonate pathway, or this methyl erythritol pathway. So there's sort of different levels of specificity of these models. I guess the point though of these models is that our idea is that, or her idea really, is that they are capturing different areas of the causal space. And so for any given metabolite in the prediction set, if its causal loci overlap with the space that is captured by one of these models, it should be well predicted by that model. On the other hand, if it doesn't overlap, if the causal loci for the particular metabolite, say, doesn't overlap with its GC2 model, then it should be poorly predicted by that model. And so what you're going to have for any given metabolite is a kind of natural ranking of the accuracies of these models according to how well they capture the causal space of the metabolite. So then downstream in the prediction panel, we look at different ways of identifying which models might be good to use. The first way requires the sort of highest level of detail of annotation for these metabolites. So it's oats, and we're looking at specific secondary metabolites that are of interest in oats. And these are avenanthramides, avenocosides, and avenasins. And what you see is that compounds that have this sort of same annotation that presumably are part of the same pathway have a high correlation of the ranking of the accuracies of these models. So if you have loci where you have detailed information, you can use that as a guide for which model to use. The second approach is to use chemical structures of the metabolites. And this is sort of a terrible slide. You can't really tell anything from it, but focus just on the colors. I hope you can see the colors in the labels of this column. And basically, these are different lipids. We've clustered them hierarchically so that they are clustered together according to the similarity of the correlation of these model prediction accuracies. But the structures of these metabolites are color-coded. And what you see is that though they've been hierarchically clustered here, the different structures don't cluster together, right? The greens are not together, the blues are not together, and so on. And so that suggests that if you know the structure of a particular metabolite, and we're using this algorithm encoded in the software classifier, that's not going to help you decide which model to use. And then the last thing Lauren has done is use an annotation-free similarity of the different metabolites. That's just simply in the discovery panel, what is the genetic correlation in the amounts of this metabolite? And so she calculates the genetic correlations. She uses that big correlation matrix to do a hierarchical clustering, arbitrarily splits it out into 10 groups. And what you find is that for the most part, metabolites that are in the same hierarchically clustered group have high correlation of model accuracy with each other, and there's relatively lower correlation of model accuracy between two metabolites that are in different groups. And this is true for 7 of these 10 clusters. For the other 3, there's a little bit of a head scratcher mystery because it seems like metabolites that are not in that group have a higher correlation than metabolites that are within groups. So it's always good to have a little bit of mystery. So the take homes are from this little vignette is that this approach that we're using of many to one and prioritizing rather than estimating, it has some merit. I mean, I think it's helping us identify models that are more predictive. And then sort of interestingly, the two extremes of the amount of information that we have, the amount of biological information, are useful guides. That is, if you have very detailed biological information, you have an actual compound annotation, you're good. If you use this annotation-free method, you're good. And somewhere in between, the sort of coarse-grained structure doesn't help you all that much. Questions? All right. So the next vignette is, so this is work that Mao Huang is doing. It's on sugar kelp. Sugar kelp has a biphasic life cycle with both diploids and haploids. I'll just run through that. Whoops. The the kelp that you see along the shore is a large diploid individual. It has reproductive tissue, sorus. From there, you can express meiospores that can be sorted into 96-well plates. These can grow up into gametophytes, which are haploid, so diploid, haploid. They are either male or female. You can increase the amount of tissue of any given haploid just by breaking up these clumps, and they regrow. And you can cross them simply by mixing vials that have a female and a male together. So these then fuse, individuals fuse, or actually the male produces a sperm cell that goes and fertilizes the ovum in the female. You get juvenile sporophytes, you spray those onto string, you wrap the string around ropes that are in these arrays that are offshore. And now you again have a diploid, and you can measure its growth and so on. You can evaluate the value of this diploid for whatever purpose, food, bioenergy, and so on. But the point is that we evaluate these diploids, but our breeding germplasm are these haploids. Okay, we select among the haploids. We grow out and we just make decisions about crossing the haploids. So we need to be able to share information between the diploids and the haploids. We want to predict the breeding values of the haploids. We have information on the diploids. It's not unlike what happens in dairy cattle. Folks evaluate cows, what's their milk yield, but most of the selection in dairy cattle happens on bulls. You can't measure milk yield on a bull. And so you need to be able to share information between cows and bulls. We need to be able to share information between diploids and haploids. So how do you do that? Nice little bit of quantitative genetics here. You start out with a co-ancestry matrix, and you want to conceptualize both the gametophytes and the sporophytes in terms of their haploid content. So I have 4 individuals, 2 gametophytes and 2 sporophytes. The sporophytes take up 2 columns and 2 rows because they're diploid. The gametophytes take up just 1 of each. You have 1s on the diagonal, so it's a co-ancestry matrix of probabilities. You sample the same allele from a haplotype twice, you're always going to get the same allele, probability of 1 that you get the same allele. So now imagine that this sporophyte, one of its parents is this first gametophyte. So this haploid is actually exactly the same as this haploid, and all of the values in this row should be the same as the values in this row. Okay, pretty straightforward. The second parent for this sporophyte is some other gametophyte, and I'm going to imagine that we know through pedigree or whatever that it has some probability 0.2 of being related to this gametophyte. So anyway, so that generates another row. Second sporophyte, I'm going to imagine it's also a progeny of this gametophyte and some other gametophyte that's completely unrelated. So, co-ancestry probabilities of 0. And then this gametophyte, I'm going to imagine is the progeny of this sporophyte. And so to get the values in this row, you just average out every cell or every column in the 2 rows for the sporophyte. Because Mendelian segregation, there's probability of 1/2 that it got this haplotype and probably 1/2 it got that one. Okay, so that's nice. That tells you how to make a co-ancestry matrix, but it's not immediately helpful because we have one observation for a given sporophyte. We don't have 2 observations, right? We'd need an observation of somehow the value of each haplotype here, but we just have 1 observation, right? So what do? You do this highly technical thing of condensing the co-ancestry matrix into a haploid related additive relationship matrix, haploid reference additive relationship matrix. And you do that just by summing the values that are in a particular cell. So 1 and 0.2 is 1.2, all this together is 2.4, et cetera. And this gives you what I call a haploid reference additive relationship matrix. If you're thinking back to your quantitative genetics and a typical additive relationship matrix, the maximum value in a typical additive relationship matrix is 2. Here I have a value of 2.4, so it's a little different. What is that difference? The difference is that in a standard diploid additive relationship matrix, you're using breeding values, the variance among breeding values, in other words, the variance among diploid additive effects, as the constant of proportionality, right? To actually model the distribution of these breeding values, you need to multiply the additive relationship matrix by a constant of proportionality. Typically, with a diploid reference additive relationship matrix, we're using these diploid breeding values. Here, I'm using the variance of allelic effects because I'm interested in the gametophytes, and they just have single alleles. And so, you know, the variance of the single alleles is about 1/2 of the variance of the diploid, or is 1/2 of the variance of the diploid breeding values. And so the haploid reference additive relationship matrix has to be twice as big as the diploid reference additive relationship matrix. On the other hand, we are evaluating sporophytes. And so if we want to get a standard idea of the narrow sense heritability of those sporophytes, we take this constant proportionality and double it in the sort of standard formula. Okay, so simple take-home message for this little part of the talk is that it's possible to predict breeding values of haploids on the basis of evaluations from diploids. And that's great because it's going to enable us to do sugar kelp breeding. Good. So now that you're thinking about haploids and diploids, I'm on to mate selection. So choosing which individual is going to provide the pollen parent haploid and which individual is going to provide the seed parent haploid. And this is sort of masterwork of Marnin in his waning days as part of the Next Gen Cassava project. So starting from the very beginning, breeding and how breeding works. We have 2 components to breeding. We have population improvement. This is recurrent. You're year after year making crosses that seek to move the distribution of the phenotypes or the genetic values of your population in a certain direction that you've decided is superior. And then you have you have variety development from any given population where, you know, you're just interested in identifying the tails. For the recurrent selection part, the only thing that matters is the added genetic variance. For the variety development, you know, any source of overall total genetic value variance is going to contribute. So in particular, single locus dominance variance, which is what I'm going to be dealing with here. I'm casting this in what we now think of as the 2-part strategy of a breeding program, where you have recurrent selection more and more frequently driven by genomic predictions to accelerate the breeding cycle. And from any given point in this recurrent selection, you spin off new variety candidates, and they go into field testing, different stages of a variety development pipeline. You get phenotypes from there. Those phenotypes are important information for reparametrizing the recurrent selection or the prediction models. Okay, in the recurrent selection part, each cross is an investment, right? You know, time, land, money. And the thing that we're interested in is just identifying the tails among the progeny of a particular cross.
Call from consumer Rob Spicer.
Okay. I'm not sure how to mute folks, but okay. So big investment. The only thing we care about is this tail, right? We want this tail as far out as possible. And I can't advance, but okay. Let's see if we can get back to. Are we back to? Yes. All right. Thank you, Moya. Okay. So people have thought about this for a long time. You know, breeders have been around thinking quantitative thoughts for a long time. So back in the 70s in Germany, they developed this idea of a usefulness criterion. The usefulness of a cross, it's not just the mean value for that cross, it's what you expect when you go out on the tail of that cross. So parameters in addition to the mean that you need are, well, for breeding values, you need the added genetic variance or standard deviation, and you also need to have some sense of the selection intensity. And so now in assessing a cross, we're not just interested in the mean, we're interested in how variable are its progeny, right? So there's some quantitative genetics to figuring that out. But note that consistent with my two-part strategy here, I have two different kinds of usefulness. The one kind just involves additive effects, breeding values, and additive genetic variances. But for varieties, it's the total genetic variance that matters. So I need a sense of non-additive effects that go into the mean and non-additive effects that go into the variance of total genetic values. So how does this work sort of from a quantitative genetic perspective? First, I'm thinking about parent one, and I'm worried just about breeding values for parent one. And I have to know its phasing. The reason I need to know its phasing is, well, you'll see in a moment, it's not necessarily relevant to the breeding value, the mean breeding value. But okay, so I have it phased, and I can calculate the mean breeding value if I know the additive effects, and now I'm dividing them by 2 because I'm just interested in, this is a mean breeding value for a gamete that comes from this parent, right? For the variance among the gametes, the breeding values of the gametes, what I need to know is the linkage disequilibrium between the loci that are contributing to the breeding value. Because so linkage disequilibrium is actually a covariance. The variance of a sum is the sum of the variances plus the sum of covariances. So I need to have a sense of the covariances among all the loci. So these are 2 different chromosomes. Nicely, among gametes, there's no covariance among loci on different chromosomes. Likewise, LD is very well behaved. There's almost no covariance between extremes on the chromosome, and it gets high covariance when you have loci that are close together. Turns out there's a very nice formula. If you call this disequilibrium matrix D1 for parent 1, and you have the value of the effects, it's alpha transpose D1 alpha. Okay, so that gives me the variance of the effects of the gametes that are produced by this parent. Now, I can move on to the second parent, and conveniently, this is why we love additive genetic effects. You can treat the second parent completely independently of the first parent, do all the same stuff, phasing, linkage disequilibrium, and so on. This disequilibrium matrix is different from this one because the allelic content is different in this parent. It gets more complicated for the dominance deviations. You can't know what the dominance deviations are going to be from one parent independently of another because it depends on the joint inheritance. So you can get a mean total genetic value from the probability of being a homozygote versus the probability of being a heterozygote. Here, these are dominance deviations. You can get a variance for the dominance deviations with this somewhat more complicated formula that requires knowing both of the linkage disequilibrium covariance matrices. But in the end, you have a variance for the total genetic values. This is just still single locus. You have the sum of the variances from the 2 gametes and the variance that's due to the interaction of the gametes. All right, so what does this give you? Sort of take homes from mate selection. The first thing that it gives you is better breeding decision support. So typically, what we've always done is just ranked different candidates based on their prediction. But then the breeder is still left trying to decide which candidates to cross with each other. Whereas now we're actually going to be able to give, sets of crosses and how good we think they are and approximately how many offspring you should want to try to get out of a particular cross. So the breeder, has really good instructions going into the crossing nursery. The other thing that we hope to get is greater gain from selection. So here's a figure from Marnin's paper where we have 306 parents that exist out in the field in Nigeria, in Ibadan. You can run these analyses on many different pairwise processes, right? 306 times 305 divided by 2, something along those lines. And so this is a figure that shows the relationship of the usefulness for breeding value and the usefulness for total genetic value for all of these crosses. There's so many crosses that the dots would be overwhelming, so it just has a contour here. And it's contrasted to 362 crosses that have been previously made by the breeding program. And what you can see is, okay, so we're very hopeful, but the best crosses that come from this prediction, are a good deal out ahead of the crosses that we've already attempted. Now, of course, here we're evaluating many crosses, and that means and we're picking out the best ones. So anytime there's error associated with the prediction, and there is certainly in this case, and you exert very strong selection, lots of what you're selecting on is the error that you generated in your analysis, okay? So as soon as we actually make these crosses and look at their progeny, we're going to be disappointed relative to how good this looks here. But still, we have some reason to believe that this analysis is going to come out ahead for us. Questions? Yeah. Yeah. So yellow is all prediction. Empirical ones are these other colors, and the purple versus the blue versus the green are different years in which they were made. Yeah, and actually the triangles are selfs, the circles are crosses. The selfs, interestingly, of course, have just as high breeding value as the crosses, but they have lower total genetic value because cassava suffers from inbreeding depression. Okay, so the last bit is Bayesian optimization of breeding schemes. And I've actually done this with a graduate student of Peter Fraser's. I know Peter Fraser is the guy who's been doing the COVID modeling for Cornell. Turns out he's a runner, so I've gone running with him. And so I got to talk to him and convince him that this would be a fun thing for him to do. I had emailed him once a long time ago and he didn't get back to me, but with your buddies like that, it works. So he put it out to his lab, and his graduate student, Raul Estudillo, has been helping me sort of figure out the software that runs Bayesian optimization. So why do we need this? Why do we need some fancy optimization approach? Analytical models don't exist that I know of. So just the sort of simple scenario, variety development pipeline. You start out with variety candidates. You want the best varieties possible. This you can analyze more or less analytically. Decide how you want to allocate your budget to early, mid, late stage evaluations. And then in principle, you take these best varieties and you cross them to start over again. But of course, we know that response to selection per unit time depends on how long the breeding cycle is, and this represents a long breeding cycle. So what you would rather do is take new individuals that are just somewhere in the middle here. You haven't spent as much time evaluating and cross them. But of course, that means that these individuals that you're going to cross, they now have to fulfill two things. Just as before, you're trying to select them to create the best varieties. But at the same time, you have to maximize the improvement of parents. Whereas for these individuals, you're still just doing the one thing. So in green, you're trying to fulfill two requirements. In white, just one. And that suggests that the green is more important, which suggests that you might want to change the geometry of the overall pipeline. And I don't know how to do that analytically, how to optimize this analytically. So, so what does Bayesian optimization do? It takes a continuous function. And so like the gain from selection over a certain number of years is a continuous function of the parameters, the allocation parameters of your breeding program. It's useful when evaluating this function is very expensive. So in my case, I'm simulating a breeding program. If I simulate a cartoon breeding program, I can do it in minutes. If I simulate a realistic size one, it takes hours. So expensive to evaluate. You don't observe any derivatives of f. So derivatives are useful for deciding how the slope is and how to go upslope. And the last thing is often you observe F with noise. So in our case, when you simulate realistically this whole thing, you have errors associated with evaluating the individuals, you have random Mendelian segregation, you have any number of things that cause one simulation to come out differently from another simulation, even if you input the same initial parameters. So my parameters are budget allocation. So on the one hand, I have how much do I want to spend on the different stages of the variety development pipeline. On the other hand, I have how much do I want to allocate to making crosses and running, genotyping all of these individuals in the population improvement side. Okay, so just thinking again about this and the problem that Bayesian optimization solves, I've run my my complicated simulation, lengthy simulation, a number of parameter settings, and the problem is I can't decide which direction is up from this. I can't decide which direction to go in. So where do I try next? If I can't decide where to try next, one possibility would be to explore the whole space and then smooth it out and pick some spot in the optimization space. But in the simple cases I'm going to show you, I have 4 parameters. If I do sort of a complete grid search of that space, it's really a lot of function evaluations. So not really feasible. So so this is the problem that Bayesian optimization solves, is given that I've evaluated the thing a few times, where should I look next? So I don't have to do a complete grid search. So here's how it works. The trick, in the smart part, clever about this is that, okay, I have this lengthy, complicated model. I'm going to develop a surrogate model. And the way it's going to work is it's going to model the outputs of the function, the true function, as a multivariate normal. So it's very flexible and fast. So now I'm going to run the breeding simulation a few times. I'm going to use the outputs that I get from this to parameterize my surrogate model. I'm going to use the surrogate to decide where to evaluate next. The technical term is I'm going to use the surrogate to acquire the next evaluation point. This is called an acquisition function. And then evaluate F there. Now I have some new values to use to parameterize the surrogate. And I iterate. And then I'm going to quit when I think I've stabilized at some maximum. So just real quick, go ahead and let your eyes glaze over if you want. But this is, so I said the surrogate model is multivariate normal. So imagine I've evaluated F twice. There's a covariance between these 2 evaluations. It's given by this function sigma naught. And here's one possibility for that function. And basically what it does is it looks at the distance between the parameter values. And if the parameter values are far apart, it gives a covariance that's close to 0. If the parameter values are close together, it gives a covariance close to 1. And it's actually, for those of you who are familiar with genomic prediction models, it's actually quite similar to the reproducing kernel Hilbert space regression model. So familiar territory in some ways. All right. So the first problem I wanted to attack was how many times do I have to run my simulation model? I did this with a small scheme. You can sort of see some parameters in here. I'm using cassava terminology, so seedling nursery, clonal evaluation trial, preliminary yield trial. And then I send the best individuals, or the individual I think are the best, to my marketing department, which is the nationally coordinating trial. I always send 4 individuals over there. So this is a sort of toy breeding scheme. And then on the recurrent selection side, I have, I'm imagining that I'm doing 2 cycles per year of recurrent selection. Each cycle has 100 progeny. These are the, these are the error variances related to the genetic variances, so that you have a heritability of about 0.1, about 0.2, and about 0.3 as you move through the variety development cycle. The other thing is costs matter here, because I'm optimizing for cost, for a budget. The seedling nursery is where I genotype individuals, so growing the seedling itself isn't so expensive, but I want to genotype it. So what I'm going to do is I'm going to start with a given set of founders. I'm going to run independent Bayesian optimizations. And when the optimizations agree with each other, I'm going to say, okay, I've reached a place where, yeah, the models have stabilized. No color. Wow, that's going to be a problem if it's like this in further slides. But okay, so the way this is supposed to look is okay. So I have actually 4 budget parameters that I'm interested in optimizing. I have the population improvement cycle, the seedling nursery, and then the later evaluation stages. And so any point in this triangle sums to 1 or to 100, 100%. And what it's going to show you is, well, there should be yellow for high gain and all the way down to purple for low gain. This did not, you can ask Lauren, this did not look like this yesterday. What a bummer. Okay, I don't know what happened. Okay, all right, whatever. Okay, so what I did here was I had 3 different founder populations, and I set off my 2 independent Bayesian optimizations on each founder population. So founder populations are in columns, and what I noticed after 1,500 iterations was, this triangle looked more like this one, and now they all look really similar. This triangle would look more like this one than it would look like this one, and this one would look more like this one than it would look. So basically, the thing that I discovered was that this is going to be interesting. Okay, so it doesn't show the colors, but it shows the red points, which are the maxima from each. What I discovered was that the overall landscape of gain, I call these triangles landscapes of gain, was similar, but that the maxima didn't necessarily, fit together. Basically, because the fitness or the gain landscape is fairly flat at the maximum. And so, here they match pretty well, but here not so much. And again, here not so much, like only 30% allocated to the population improvement cycle versus 42%. So the shape of the landscape would match, but the maxima didn't. Okay. Well, it looks something like this. And so I sort of invented a metric that showed the difference, the distance between two gain landscapes. So here's one gain landscape, here's another one, and I can calculate a distance between these two. So if I start with different founders and I iterate my Bayesian optimization, pretty soon it's putting together gain landscapes that are similar, but eventually that stops. And then interestingly, if you start with the same founder, 2 different Bayesian optimizations, it continues to sort of fit aspects that are particular to the set of founders that you started with. But so I figure, since we don't actually know what the characteristics of the founders are that we have, I'm thinking about 200 iterations is good enough when the landscapes that started from different founders start to look similar. Okay, next question. I mentioned that if you're just trying to optimize the variety development pipeline, there's quick analytical solutions for this. And so what I wanted to know is, if I optimize this VDP in the full context of a 2-part strategy, how much does it look like the optimization from an analytical approach of the VDP, just considered in isolation. And so here I'm doing this on a more realistically sized breeding program where the simulation takes 2 or 3 hours to run. So I start with many more seedlings and so on. Otherwise, everything is the same, and the recurrent selection is cruising along with quite a bit of effort, lots of progeny. And okay, well, I'll just show you where the maxima are. And interestingly, okay, so they're pretty different, right? When you optimize for the 2-part strategy, which are these 4 somewhat different triangles, you allocate a lot to the seedling nursery where you're genotyping. You allocate almost nothing to this the final stage of evaluation, where you know, in the final stage of evaluation is quite distantly related to the individuals that are in the recurrent selection cycle. On the other hand, you have to invest a fair amount in that final evaluation if you're just considering optimality for the variety development cycle considered on its own. Okay. Yeah. Yeah, so this is a percentage of the budget that's going into the VDP. So this adds up to 100 and this adds up to 100. Okay, and then the last thing I wanted to look at was differences between a short-term gain versus long-term gain. And the basic idea here is over time you have your population that's improving, but periodically you pull a set of lines out and you do variety development on them. And so if you have a short time frame, the overall gain, so you start from here and then you end up here, the overall gain is dominated by the variety development pipeline. You look at long-term gain, again, I'm starting from here, now I'm going all the way to here. Here, the difference is dominated by how well my population improvement goes, okay? So that would suggest that you need to pay more attention to population improvement if you want high long-term gain than if you want just short-term gain, right? And this is the saddest thing. Okay, so anyway, so my short-term gain was 6 years, my long-term gain was 12. I feel it. So many people for long-term gain would go longer than 12. Philosophically, I think 12 years is way beyond how long you would use a single breeding scheme or single breeding approach. So what's the point of optimizing a budget if you're going to change your breeding approach halfway? But what tickled me, what I think is really fun, is that actually what happened was the Bayesian optimization is suggesting that I allocate more budget for the short-term gain and less budget for the long-term gain, or actually more correctly, that when I want high long-term gain, I need to invest a lot in the phenotypes, because the phenotypes at the end of the day are still the quantum of information that generates accuracy for your genomic prediction model. If you don't have the phenotypes, your genomic prediction model, what it's going to do here is it's going to run through a lot of the variability, and there won't be variability left for long term. Here, if you invest in the phenotyping, you're maintaining a higher level of accuracy, and you're reducing the loss of genetic diversity. So interestingly, even though what I want to do is prioritize in this long term the population improvement cycle, what that suggests is that I have to invest in the thing that makes the population improvement cycle work, and those are the phenotypes. Okay, so take homes. First of all, interestingly, the founding population itself has some characteristics that affect optimal allocation. I'm not sure there's much we can do with that because you know, I just don't know what aspects of the founder population matter or how to measure them in practice. I think about 200 simulations should be good enough. I mean, that'll vary from scheme to scheme, but it's an order of magnitude. Analyses that optimize the VDP alone, you can do them analytically, they're fast, but they don't necessarily give a good guide. And interestingly, maximizing for long-term gain requires more phenotyping because you want to maintain the accuracy of your genomic prediction model. And usually on my Mac anyway, it generates pretty pictures, but not so much here. All right, and that's everything I have to tell you. And so I think this is probably the end of our time. I'm not quite sure when is it supposed to end? But so if folks need to leave, I know that there's 4080 is happening over in the field house. So, huh? 115? Okay. Oh. Okay. Well, anyway, so we have some time for questions. Well, right. I mean, I think typically, right. You would use the set of founders that have some level of diversity, where also you expect you know, positive dominance deviations in crosses among those founders. Yeah, you can try to select out those founders. I think even if you did that, there would be differences between different sets that have something to do, say, with the level of LD between causal loci that you can't measure because you don't know where those causal loci are. And I think those types of things affect the variance in ways that the optimization picks up on, but that you can't get a handle on initially. Well, repeating the question, right? So selection intensity isn't the only determinant. The number of individuals that you're looking at matters because it changes the probability that you're going to identify a superior segregant. And well, I would say my complex function that the Bayesian optimization is working with, models all that. It models random Mendelian segregation. It models random recombination breakpoints. It models linkage disequilibrium among loci. So yeah, one way or another, it's going to take that into account. And that's why it takes, 3 hours to run one simulation, and why I want to minimize the number of simulations I have to do to get a sense of my budget allocation. So it'll the major challenge, it turns out, for using this Bayesian optimization, at least for me, is that it's in Python, and I'm comfortable with R, and getting R and Python to work together is not easy. So my joke about this is that the Bayesian optimization, it optimizes a function that's a black box. So you have this Python thing that's querying an R-based black box. But the black box accounts for all of these things that you're thinking about, Mark. Will? That's a good question. I haven't actually, this is hot off the presses. So I haven't talked about it with folks in Nigeria. I think one of the important features to remember about this is that at the maximum, you have a fairly flat gain landscape. So there's options for doing a lot of different things and still having it work pretty darn well. And I sort of liken this to yield components, where you might yield components, you estimate in having more tillers per unit area, but then you have fewer grain heads per tiller, or fewer seeds per tiller, something like that. Likewise here, if you invest more early, you invest less late, but invest more, whatever, you still have, what matters ultimately is your budget. And there are optima to be found, and I think there are sort of take-home messages about what not to do. Like in particular, you don't want to invest too little in the stage where you do the genotyping, because the genotyping stage is what feeds your prediction model, right? If you have a small, if you have a small set of individuals who are genotypes, you can't have high accuracy. Likewise, one of these things showed, if you invest a ton in the population improvement cycle, that means that you have very little in your phenotyping, and it crashes sort of immediately. These are like, but anyway, the Bayesian optimization picks up on that. Yeah. Okay, you indicated that the optimum for breeding progress was fairly flat. Yeah. Do you think that holds up? If so, progress will be pretty robust to changes in student talents, grant funding, non-funding, and other unmodeled factors. Okay, that's pretty interesting. Well, okay, so grant funding or non-funding, that doesn't work, because a flat model at the top doesn't mean that you can take away from the budget and still have it work, right? So then student talents, I'm looking at a very narrow set of parameters here. I'm looking at how much to invest in the different stages, and I'm not looking at how carefully the student is doing their work, or whether indeed you're doing a good job of trying to select for traits that have heritability, as opposed to investing a lot of selection effort in traits that you would really like to have, but they have very low heritability. If something has very low heritability, it doesn't matter how important it is to you, it's not going to help you to invest a lot of effort in it. So thinking along those lines, I think, now at the same time, my statement, I'll defend it by saying, if you go out there and you look at the world of wheat breeding, wheat breeders do a lot of different things, right? they don't all have the same breeding program. And so I think part of the reason why that still seems to work is because, yeah, a lot of different things work. You know, I'm sure there's an optimum, and that's determined here at Cornell. Vote for me. But anyway, a lot of things work.
Any other questions?
Yeah. Yeah, I think you could do that. That's sort of interesting. So for example, in the cassava project, we've invested a fair amount of effort in developing near-infrared spectroscopy. So that's a technological innovation. What the technological innovation does is it allows you to phenotype more individuals somewhat less accurately, right? Is that an allocation that's a good idea or not? And what's cool about this Bayesian optimization is it's optimizing a black box. It doesn't care what the parameters are. So you know, I mean, you don't want to have 30 different parameters that you're trying to optimize. I started with 4. I'm sure I can go up to, you know, 7 or 8 or 9. And I think those ideas, like so Marnin might use this in the mate selection. Like, how much are you going to emphasize usefulness for breeding value versus usefulness for total genetic value? Or, you know, another thing that matters a lot in long-term selection versus short-term selection is, what is the effective population size that you maintain in your recurrent selection population? So you can add that as a parameter. Right. So yeah, I mean, that's one of the cool things about this approach, is that I think it's robust to lots of different possibilities. I think there was, yeah, Carl. Well, yeah. I mean, I think the black box that you optimize, you can make that black box as complicated as you want to. You know, there's trade-offs in that, right? The rule of thumb for models is you want the model to be as simple as possible, as long as it captures, you know, the phenomenon of interest. So make it only as complicated as it needs to be to capture the interaction that you care about, and no more complicated. So for example, I started out by programming a simulation model that included checks. And it turns out adding checks to a simulation model is kind of a pain in the neck, and it makes it a lot more complicated. I mean, it's not a huge complication, but in hindsight, I was like, well, you know, the checks, we're not really using them in assessing in the simulation. In the simulation, I don't simulate any kind of spatial variability or whatever, because, you know, whatever. This is a long discussion, but yeah, that was a wasted effort. And in reality, you need checks. Yeah, I'm not disputing that. And maybe, you know, if you wanted to start to model spatial variability or more complex G by E interactions, where you might be able to assess something about the environment based on the checks that you had, yeah, then you would want to put checks in there, because it would allow you to do that. But otherwise, don't.
This has been a production of Cornell University, on the web at cornell.edu.